Welcome back, everybody, to our session on basics of cell signaling. So, how can our cells communicate? How can they exchange information? They can do so via so-called cell junctions. They can also communicate via cell-to-cell recognition, and this type of cellular communication is quite important for our immune system. Our immune system is able to recognize, for example, if a cell underwent tumorigenic transformation and needs to be eliminated. There is also a type of cell signaling mediated by biochemical signals. And the first type of this signaling is so-called paracrine signaling. In addition to paracrine signaling, we can have autocrine signals, synaptic signals, and there is also endocrine signaling. So what are the differences between those types of cellular communication? The first two types I showed you before are communication over very, very close distances. In order to communicate via cell junctions or via cell-to-cell recognition, cells need to be adjacent to each other. They need to be very close. In contrast, paracrine, autocrine, and synaptic signaling are ways of communication over mid-ranges, because they involve secreted molecules that are released by one cell and picked up by another cell. Last type of cell communication, the endocrine signaling, usually occurs via very, very long distances and is usually mediated by hormones. Hormones can be released far away from the target organ. Hormones can be released by the brain and can be sensed by cells in the periphery. So this is communication over long distances. So let us have a quick look at cell junctions. What you can see here, and this schematic drawing of two cells, two neighboring cells, with tight junctions indicated here. Again, as I mentioned before, tight junctions only allowed communication between neighboring cells. They seal neighboring cells together and prevent leakage of molecules. They control exchange of those molecules. One type of cell junctions are so-called desmosomes, and desmosomes join intermediate filaments in one cell to those in the neighbor. Importantly, cells can actually exchange information if they are joined via the cytoskeleton. For example, if one cell senses pressure, this information is also received by the neighboring cell via the desmosomes. The last type of cell junctions are so-called gap junctions, and as the name already suggests, they are gaps between the cells, and they allow passage of small water-soluble ions and molecules. Overall, junctions direct the exchange of information between two neighboring cells. So again, this is a very close contact type of communication, and they allow exchange of ions and small molecules only. But you will hear more about cell junctions in a dedicated lecture which will be delivered by Dr. McNish. The next type of communication between cells at close range are cell-to-cell recognition. Because cells communicate by interaction between molecules on the surface. In local signaling, human cells may communicate via direct contact of membrane-bound cell surface molecules called receptors, and this is basically what you can see here. The cell on the right-hand side is sensing presence of certain molecule, the green one, present on the surface of another cell. After sensing this, the cell on the right-hand side can activate certain downstream signaling pathways. Our cells are also able to sense presence of glycoproteins, lipids. And this type of cell communication is very, very important for the immune system because, as I mentioned already before, certain cells within our immune system are able to recognize cells that are infected or have been transformed into a tumor-like cells. Once this type of cell is recognized, the immune system is able to actively remove cells that are infected or change. Good. So let us now talk very briefly about paracrine signaling, autocrine signaling, and synaptic signaling. All of those ways of communicating can be summarized as local signaling. So the information can actually pass slightly larger distances than cell junctions and cell-cell recognition. However, the range of this signaling or communication is usually fairly limited. So we have some cells that secrete molecules, and those molecules or signal can be sensed by cells adjacent to those. They can take up those molecules and react by activation of certain downstream signaling pathway, and last but not least by a certain biological function. So signal molecules are produced and secreted by the transmitting cells and then diffuse locally until they encounter target cells with appropriate receptors. A very special type of paracrine signaling is synaptic signaling. And this communication is limited to nerve cells, to neurons. Here, electrical signals along nerve cells trigger release of neurotransmitters. Those neurotransmitters can then be picked up by neighboring cells, and this leads to stimulation of the target cells. So again, synaptic signaling is paracrine signaling that occurs at the synapse structure between two neurons and is more directed than general paracrine signals. The last type of local signaling is autocrine signaling. And this is actually quite interesting because in autocrine signaling, the transmitting cell and the receiving cell is the same. A cell can send signals to themselves, for example, to amplify certain biological reaction. Right. The last type of cell communication is the so-called endocrine signaling, and this occurs over very, very long distance. Here, a transmitting cell secretes hormones in one part of the body, and those hormones are then transported to the target cell via the bloodstream. And this type of cell signaling can actually occur over very, very long distance. Again, some hormones are produced in the brain, both received in the periphery of the body. So, what are the types of cell communication in a nutshell? We have paracrine signaling, we have autocrine signaling, endocrine signaling, and there are also signals transmitted between cell via cell junctions and cell-to-cell recognition. With this, I would like to end session number 2, and I'll see you shortly in session number 3. Thank you very much.